So this morning, I gathered here for a presentation of a seminar, and we are privileged to have in our midst visitors from far and near. And we also have our faculty members, students, and postgraduate students who also wish or understand the reason why we're here. And so, in order to start this activity, we need to introduce our chairperson for this, occasion. So I read a short about our chairperson for today, and then we commence this activity. All right, so in our midst, as the chairperson, is the Dean of the School of Allied Health Sciences, who is in the person of Professor Testimony Kumale. All right, so we'll now take some remarks from our chairperson before we begin today's activity. So, Prof, that's it.
So, thank you very, very much, our guests, faculty, and the students. This is a very important seminar, which I know we will learn a lot from. So, I will invite you to pay attention, jot down your questions. After the presentation, you'll be given the opportunity to ask questions. Usually, seminars, there is the question time where the discussions actually starts. So, after the presentation, I know we have that time to discuss more. And I know during that time, most of the issues we want to actually discuss and then pave a way for the next level to come. So thank you very much. And I'll accept the chair Debbie. Thank you.
Thank you very much for accepting the chair today's meeting. To introduce our guests and our speaker for today, we'll call on Dr. Rachel Odakonos to do that.
Thank you. So before I introduce our guest, I would like to acknowledge the president who has been from Department of Anthropology, also Alexander Julius, the head of the Department of and then we have colleagues from Department of Medical Laboratories, Dr. Hidu and Dr. Listen for also have our own department, fantastic. Yes. And then Dr. Amma is okay now. We have our postgraduate students. So we have our own Professor David, Professor LaKisha T. We identified here we have two students attending the Professor LaKisha is an assistant professor of the Department of Anthropology at the University of Illinois at Urbana-Champaign. So we have the pleasure of on genetic genealogist. He has their own lab, named the Lab African Genetic Technology Lab. I think you really have from your home. So she will talk today to us about because how we can bridge the gap between those of us here and then those in the diaspora. But we want to use our own scientific tools, we have to use the other to be able to do this. So please help me welcome Professor David Patrick.
Thank you so much for your welcome, for inviting me here to speak. The Dean of the college, Professor Rachel Mills, and everyone here for giving attention to this. Please let me know if you can't hear me, because I do tend to speak low. Can you hear me? No? Louder? Okay.
I can hear you. I can hear you.
You can't hear me?
Yeah, very loud.
All right, all right. So we'll begin, and I'm not sure how to minimize this further. Okay, we'll do this. Oh, yes, of course. Everyone can see here? Okay. All right. All right. So bridging the gap, family ethnic identity development through genetics, the power of genomics, linking generations across continents. So as mentioned, I am LaKisha David, assistant professor in the Department of Anthropology and the University of Illinois at Urbana-Champaign, but also by profession, I am a genealogist. Again, thank you very much for this invitation to speak. A little bit of background. I got my master's degree at MIT at International Development Group. What is can you hear this?
Yes.
Okay. All right. So this is like the second and third time I've come to Ghana during my master's program. When I was in my undergraduate program, I spent a semester at KNUST. So I was telling Professor Mills that when I walked on campus, it sort of reminded me of those days. Then I went on to get my PhD in human development and family studies, where much of the talk today will be based off of research that began there. And then I went on to the University of Pennsylvania for my postdoc training in the ethical, legal, and social implications of genetics and genomics. And then here. So in any event, I remember in those days when I was sitting in a very large classroom. So I feel like I'm on the side of undergraduates here. So if there's anything you need me to tell your professors, just tell me on the side and I'll pass that along. All right. while we're waiting on that, so have you heard of people taking the DNA tests in the US and other places and getting their ancestry information back? You've seen these like YouTube videos? Someone's seen it, right? Yeah, like Erica Badu. What have you thought of those? Thank you. Interesting. Not really. Kinda. What is it about? Okay, so we're gonna talk a little bit about that. All right, so as an overview, this is my research program, but I want to go back here. So I'm going to talk about, just give an overview of 3 of the projects that I'm working on. The first one being genetic genealogy inference, where we're using genomics to try to infer a multi-generational family tree. And then really the bulk of the study is on the social impacts of this, both from a diaspora standpoint and an African standpoint. And then open it up for discussions. I will allow just, some clarifying questions along the way, because I want to make sure everyone is with me in this. But as the dean mentioned, the bulk of our main discussion will be at the end. So we'll go back to this picture here. This is the research program in a nutshell. So the person here is African American. She tested in the project and learned that she was related to people in the northern region of Ghana. So here, if you're familiar, this is Paga, the Goro slave camp here. And people there have tested in my project. And so this lady, after learning that her relatives were there, she traveled there to visit with them. And you can see the expression on her face, what it means to her. So in the first project, I go back to the commercial DNA test, where oftentimes, this one is actually mine from 23andMe, where they have the 1 to 22 chromosomes and then the X chromosome, 2 bars for each one, for one from each parent. And then each segment color coded based off of where they think that region of the genome came from. I do find that the way that this is displayed is very problematic. And a lot of my work is on trying to correct misinterpretations of this. So many people in the general public don't understand what a reference panel is. They don't understand that these are individuals and that this is a measure of how related you are to individuals. And that there's not very much difference between one population and another population. It's more about the allele frequencies, right? So there's a lot of work that needs to be done towards unlearning and learning the proper interpretation of this. And I run into this issue when I'm doing the work. So even though you might hear it being referred to as ethnicity estimates, this is not a phrase that I use, right? And if you're curious, what this does show is that I'm related to many people across many places in West Africa, mostly. Some indigenous US, European. Genetic ancestry, or biogeographical ancestry is a better term, but I prefer genetic similarity or genetic relatedness, because this is essentially what we're talking about. Every individual on the planet who has ever lived we're related to each other. It's just a matter of time to most recent common ancestor. So I'm going to use that phrase a lot, the time to most recent common ancestor. So that is a sort of a key point in the work. So we're talking about correlations of alleles, or that genetic similarity between individuals. And the reason why I'm stressing this is because oftentimes we tend to think of genetics in population terms. And I want you to, switch gears and thinking about it in terms of family and individuals in that sense. So we're talking about identical by descent segment sharing. And just to give you an idea, we're talking on the scale of 3 centimorgans to, 100, 200 centimorgans, which is a measure of the genomic distance. All right, so identical by descent segment is that segment of DNA that's shared between two individuals that indicates that there's a common ancestor. And just for those who might be more social science oriented, what we're talking about is this IBD segment. It's this segment here, or in both individuals, this segment is the same. So for this individual, they have inherited from their grandparents through their parents to here. And so the color here represents a recombination of the same for this up. But when we do the genetic studies, the only thing that we will see between two individuals is the IBD sharing, which will be here. And then from that, we will have to infer how they're related to each other, either through oral histories or testing more people to just based off of that information, build up how they're related to each other. Unlike in population genetics, the rest of the genome in this sense is irrelevant, right? So we're not comparing groups to groups. Once we have this information, that's the important part, and that will then direct how we continue in the study. Any just questions here? Thoughts, comments?
Are you looking for the smart one in the, you know, what I'm thinking?
Right. So I'm not looking at just one area of the genome. It's just wherever in the genome that they are similar. Did I answer your question?
Yeah.
Okay. Any other clarifying questions? Okay. All right. Is Samantha in the room? Where's Samantha? Okay. All right. So again, all individuals are related to one another by varying levels of relatedness. This is a core of the research, the lab, my thinking in this. And I oftentimes have to emphasize that. So forgive me for emphasizing it. So sometimes people will say to me, oh, but you know, Africa is the most diverse place in the world in terms of genetics. Isn't that a problem? And I want to, that's not a problem, because regardless, a parent and child are going to share, a child will share about 50% of their genome with each parent. That doesn't change, right? These principles don't change regardless of the individual population or whatever. A grandchild will share about 25% of their genome to each grandparent. First cousins, 12.5%. Doesn't matter where, like as you saw in my genome, some of my relatives are European. That, where people are from is irrelevant. We're talking about genetic relatedness. And wherever that segment happens to be, that's the part that we will focus in on. Right around the fourth cousin level, the third cousin level particularly, is where things begin to shift. So at the second cousin level, but there's a 99% chance that if you are related, it's going to show up in the DNA. But at the fourth cousin level and beyond, that's when you begin to share so much, the amount of DNA that you share is reduced that you can actually be biologically related to a fourth cousin, but that IBD relatedness does not show up in the DNA. So I can be related to you as my fourth cousin, but it does not show in our DNA, which I think is a really fascinating fact. So you can be related to a person, that's information, but not showing is not information, unless it's second cousin or greater. In other words, if you're a parent, a child, that's going to always show up. So these things is what we need to factor in when we are referring to these family trees. The fact of the matter is that we have such a large number of 4, 5, 6 cousins that we find them. And you only really need like a few to begin to piece together the family tree. This chart, I don't intend for you to study it. It's no quiz or anything to undergrads in the room. But I do want to show you that there is a science to it, even though it's not population genetics, it's family and individual genetics, there is a science to it. So empirically, people have, let me orient you to the chart here. It's like a family tree. If this is you, this has been your parent and your grandparent. It's been a sibling of your parent, aunt and uncle, and then a first cousin. First cousin one removed is a child of your cousin. And what I want to point out is that we have a percentage of DNA that we're expecting that relationship will share with you, an average amount in our range. So these numbers is what we put together when we build in our bioinformatic tools. These probabilities, or statistics really, frequencies. But there's two things to take into account is that once you get to maybe second cousin or so forth, that range overlaps. So you can share, let's say 50 centimorgans with someone, and it could be a possible 5 or 6 different relationship types. So that's part of the challenge of where that person fit in the family tree. In which case you would then have to test more people and try to find out where they fit in the family tree. Get me? Get me? Sure. All right. I am a teacher, so I you know, a little feedback is good. All right. So the main objective here is simply to explore the genetic relatedness between Africans and people of African descent. I am focusing on the families that were separated during the transatlantic slave trade. In new framing, we will say mass human trafficking from Africa to acknowledge that these were people and not property in you know, that sense, or objects. My focus is on Ghana and US residents and trying to find those relationships. Did anybody think about this question? Wasn't it too long ago? No one was curious about that? Interesting. How about, well, isn't, you know, slavery so long ago? Why are you still talking about it? Okay, you're not gonna tell me right now. Okay, that's fine. But it wasn't that long ago. So this one is the slave voyage is a really reputable database. They have collected manifests from slave ships across the Atlantic and put together this database. So we know with some statistics about how many people were trafficked from Africa to various diasporic locations. Not that I need to show you in this room, but you know, we're talking about here to various places across the world. This diagram is that same information, but just in a timeline sense. So you have the time, 1500 to 1850 here. You can see the number of people who were trafficked during that time. The difference here is, if I'm not mistaken, more or less people who died along the way. And the reason why I show this is because, again, really speaking to population genetics, oh, it's too far, you know, too long ago, the time frame is too long ago. And I respect the question, but in actuality, it wasn't that long ago. If we're looking at 1866, 158 years ago, let's say on average, 25 years per generation, we're looking at about six generations ago is where it ended. So yes, you know, 400 years, maybe relatively a long time ago, relatively speaking, but we're talking about, you know, the latter part of it and the legacies that is with us today. So a person who was born in like 1960, this is only like 4 generations ago. Okay. Can we detect DNA relatedness at that level? I need some nods, because I know, you know, yes, right? It shouldn't be a question. Does it matter that it's, you know, African populations? Not at all, right? Okay. So then I focus on 1800 because this tends to be the range the higher range where we are finding the relatedness from 8 to 18. 8 is artificial minimum. 8 is where the commercial companies that's their cutoff. But we tend to see around 18 to 20 max. So I use this number here. That's only about 8 generations ago. We can be conservative and say 10 or so forth, but the point is, it's not that long ago that we can't pick up this relatedness. So I'm literally talking about that, like, you could be my cousin from, with our shared ancestor being maybe, let's say, 5 generations ago, right? And I want to share with you what that means to people of African descent, and hopefully to learn more about what that may mean to Ghanaians. With the work, what I'm hoping to do right now, we have an algorithm that does well up to 7 or 8 generations. And my graduate student here, Mackenzie, and I are working on probably using machine learning to expand that. So that's the work that we're doing. Oh, I should also add Samantha soon, hopefully, and Dr. Mills, of course. There has been work in this area that, you know, gives us some hope as well, even though we know in this room. So this one was done by 23andMe. And you can imagine how large that database is. And so they took a sample of the research, people who consented to participate in research, and then other data. And they looked at all the individuals that shared at least 5 centimorgans of DNA. And if you're a population geneticist, you know that 5 is a considerable amount to share. What was interesting to me was that 88 to 91% of the diaspora had at least one IBD-related IBD segment with an African person in the database. So for most folks, we would probably find at least some related information that we can then explore further. So if you're Gold Coast here, we're looking at about 15.5 is the peak, telling the most recent common ancestor. But again, you know, this is also based off of the sample they use. They tend to use folks from the coast, whereas the research that I'll be talking about is across Ghana. Any thoughts, questions really quickly before I move on from here? Okay. Any of this surprise you? All right. So let's go into a real case here. This is Eileen. She's African American, by the way. I'm using names that I've been given permission to use. So this is Eileen. She's African American. And Rhoda Quagreen, she's Fonzie. Her folks actually live in Elmina. And so first Eileen and Rhoda tested. And this segment here represents the part of the genome that they share. That's that IBD relatedness. You can see there's little snips, little markers on the genome where they also share, but it's not consecutive, and so we don't count it in this case. But we know from this segment, that consecutive space, that this indicates that they have a common ancestor in a, you know, measurable amount of time. And so just if you're thinking about like the bioinformatic tools, you have to start in base position to measure in centimorgans, number of SNPs, and so on. Okay. This is using GEDmatch, but my lab is working on our own tools. Looking at it a different way, so we started with Eileen and Rhoda. We tested them, we found out that they have IBD relatedness. Doing other statistics, we know that they have about 7 to 9 generations to their most recent common ancestor. So 7 to 9 times is in generation, most recent common ancestor. Okay. So for Eileen, we would test more folks in her family tree to find which parent also matched Rhoda. Now, I understand you could probably use mitochondrial and Y, well, not Y for her, but, you know, other ways, but I'll show you in a moment why the autosomal works for this. So in any event, same thing for Rhoda. We've ended up testing Rhoda's maternal grandmother, her mother as well. I think that's it for her. So this is a real case here. Okay, so that would be Rhoda's mother. And then continue to test in Rhoda's grandmother. And then at this point, we have to randomly sample people within her family tree, her extended family tree. So it's not random. It's a word that I would stutter on, so I won't say it. Purposeful sampling, that word. All right, but it's an intentional sampling of people within our family tree on her maternal grandmother's side to try to find, where that match would be. So once we do find that person, then we'll be able to build from Rhoda to that person. And generally speaking, this will be known information confirmed through the DNA. So asking, are we only focused on one part of the genome? At this point, we will focus on one part of the genome because what we're looking for is that same overlapping segment. It's okay if it extends, one side over the other, but they all must have some overlapping segment. And that will be key to putting together the tree. Same thing for Eileen. And putting together the family tree here. Mind you, we've said it was only 7 to 9 generations. Once you've gotten to this point, that number can probably be refined. But even if we were just to stop here, this is a whole lot of information that they did not have before, right? But then the work, once we're able to place this, even if we don't know names and things like that, this is putting together the tree. I understand that there are tools in population genetics, TS Infer and other bioinformatic tools, although it tend to have a longer time frame. So you're looking at 25 generations. I just want to finish my sentence real quick. So you're looking at 25 generations and beyond. It uses coalescence theory, so it makes generalizations that is not applicable when you're looking at within 25 generations. So that's why we have to develop new tools for it. Good. This is my number one thing that I do not want to deal with and that I have to deal with. So this is later, but I will, since you asked it. I did, in getting RB approval, we have to these are the risks, these like this, right? And I thought I could get away because my research doesn't focus on fraternity and things like this, right? It focuses on these distant, family trees, but clearly I have to, understand more local, closer relatedness to even build the more distant in a way that I would like to. And we did find this where the mother passed away. We tested the son and we tested his father. They're, biologically speaking, they're not, father and son, although socially they are. But not only did my RB not cover that situation, and they consented and did not say that I can share that information with them. There's two things. One, the African Americans who are related to them are going to notice that they're related to one and not the other. So I can't just hide that information. And the other is that the son depends on his father to take care of him. And the mother is not here. How can I disrupt that? So this is a question I present actually to the people in this room and, how do I deal with it? And then also I understand that local governance, royalty laws and things like that is based off of these lineages. What happens then, So this is a question and I'm glad you brought it up because I don't really know how to handle it.
Don't know. I think the simplest way to go is to maybe trees through feminine. That one, it's but the moment you bring, there will always be.
That's, I mean, okay, we'll put a pin in that if you don't mind. It will come back to it. All right. So this is a real family here. Real family. What I mean is this happened here. This lady here, Rhoda, this is her mother, and she lives in Kosovo. To the right, to the left of Bakra.
Sure.
Yes, that's where she lives. And then her uncle is here somewhere. So it's a blended family. He's African American. This is Rhoda's sister here, and so forth. So this is her father. So my point here, and then her grandmother, I don't see her in this picture, but lives, as I mentioned, in Elmina. And so the African American cousins traveled here to Ghana to meet with them and this whole thing. And then this one is in the US. This one here is, he's Ghanaian and his mother. This is Sherry, African American, and his son. These are people in my project. They tested, found that they're related to each other. They thought that she was related on the father's or the mother's side. Turns out she's related on his father's side through additional testing and so forth. But this is on the outskirts of Chicago. They meet, they dance, they have food, you know, they meet regularly, and they're becoming family for what it means to them. All right, can we go on? All right. So I mentioned the Ghana project. So it began in the north. It began in Pikworo slave camp here in the background of this image. In Paga, and these are some of the first folks that helping me with the proof of concept. This is Gabriel here. In the middle there is Regina, and these are some of the elderly folks who tested first. This is out when I was a graduate student, trying to prove to my professor that this was a thing. And so kind of went rogue a little bit so that I can show them. So in any event, more here. And eventually we got to a point. He has the Illinois shirt on. It was a gift from my professor at the time to him. But you can see at this point, fast forward in it, he's now trained to like administer, you know, the DNA test, to do like demographic collection and so forth. And this is John here in the yellow shirt. Just so you know, when I'm in the US and I'm, you know, I have to show them where Paga is and everything, but I don't think I need to do that here, right? Okay, I was gonna sit, never mind. Okay, so here's a joke that I had in mind. I often tell Ghanaians, like, I've been to more places in Ghana than Ghanaians have, so like, stop playing with me, you know, anyway. So yeah, so 35 participants, we were tested more at this time, but this is the core sample. This abstract was accepted in the Ghana Studies journal that Professor Mills and I, and along with my team, we're gonna finish that manuscript up. So we tested 35 folks at the first stage. Here, again, another person who was trained and helped with translation work and things like that. This is Gabriel collecting some of the demographic information, history information. Okay. So back into the measuring the genetics. So just among them, they shared on average almost about 300 centimorgans, which for me is a good thing. Whereas oftentimes in GWAS studies, you want to remove close relatives. For this study, we need to have close relatives. And so the denser you test within a family tree, an extended family tree, the better. It's, you know, making more accurate. So just based off of IBD relatedness with each individual in that sample, we found 9 family groups. Again, just speaking biologically speaking, because family is more than genetics. So them in the Ancestry DNA database, as of April 12th, they had 6,000 unique matches in the database. And to me, I was so excited. And it was like, see, like this is the thing, this is happening. The vast majority of these will be people of African descent, African Americans, Jamaicans, and so forth, just based off of where the testing happens. Most of these, well, I'll show you here. When I reduce this group of matches to just the highest match they had among each pair, the average sharing was 11 centimorgans. And so that's about 13, 14 generations to most recent common ancestor, which now that I'm thinking about it, is really similar to the other one we saw from the 23andMe sample. All right. And again, the 8 centimorgan minimum is just an artifact. Thoughts, questions, or surprises? All right. Also was interested in the social aspect. I'm sorry, Professor Mills, I forgot to start my timer, so let me know if it gets to running long. Thank you. All right, so we got the social aspects. I was really curious about what this meant for Ghanaians. Also, I thought this is also an ethical thing to do, to understand what it would mean for Ghanaians. So we did focus groups before the testing, we tested, and then focus groups afterwards. I led the focus group before, and Gabriel led the focus groups after. We also stratified by sex, or you say gender, and by age. So we had older and younger, male and female. So 4 groups. And so I'll show 4 things that sort of stood out to me from the group in Paga. One of the things that stood out to me is the phrasing that they use to refer to their diaspora relatives. Siblings, brothers and sisters, children, relatives of the same blood. And I guess from my perspective, from the US, I was just thinking, okay, cousins. I also get, asked a question about, oh, 4th cousin, 5th cousin. Like, that becomes irrelevant once we find out that you're related. It's just like cousins. But when I was talking to them, they continued to refer to them as brothers and sisters and siblings, I guess much like they would a cousin here anyway. And mostly because of the elderly will refer to them as, oh, my children and things like that. This one was another phrasing that was really interesting to me, the way I just want to see them first so that we can know ourselves and greet ourselves like it is done with Casino people. This is from the younger male perspective. Okay. This is the older men's perspective. The community is happy. We found our relatives, our lives will be better, and our community will develop. And then the younger, it's not like we expect something from them, but when they come, whatever they have, they give us, and we also give them what we have. And then the elderly women, their cry is that they are from here. We also wanted to know if they are our relatives so that we can draw them home. Sort of ran through that, but I just really, I'm eager to get to the discussion part of this part. I would say that the older women tend to be more like motherly and more emotional, whereas many of the, like say the older men were more like industrial about it. They did tell us about stories of being discriminated against when they go to the feed shop. Even though they have money, people won't sell them things. And so they wanted African Americans to like stand up for them and things like that. But if I'm being real, when I first, was going up there and I took up a bunch of students, like sometimes they would ask my students, oh, can you buy me a tractor and things like that. And so, and for my students with their heart on their, on their sleeves, like, oh, welcome me. It was very like heartbreaking. And I needed some time to process it. In the US, when people are more affluent, right, those more wealthy relatives are expected, they're called on a lot to try to take care of the ones who are not doing as well. And I understand that the same here in Ghana. Someone goes to Accra, something like that, they're like really relied on, to like maybe, bring on another person or something like that. So in a lot of ways, as I'm explaining to, folks in the US, and hope you can help me to understand it better, it's really similar. And I can't say that it's just about financial gain. Because at the same time, if a diaspora person go there, they're given land and things that we, generally speaking, African Americans can't afford in the US. And so it's like, I saw the land they were giving me and they were giving others. It was like, this is just a normal thing you do to family members, for males, But anyway, we won't go there too much. But, there were certain privileges made for me. So I do explain that it is sort of this reciprocity, in that sense. I think it's really real. I think that I need to spend more time to get, the really true, more truer stories. And that has happened. They've revealed more things to me since, collecting this as data. So some of it doesn't make it to data, but they make it into, my personal relationships with them. The other thing, though, is trying to help people in Ghana to understand the riches that you're pouring on to a person of African descent when you say, welcome home. And this is your home, right? That's more than like, overcharging for my taxi ride, like on the next sentence. There's a wealth to that I will not be able to explain to you. Yeah, so like you have like grown men who will cry because of it. You get me? Like it is a richness that you're actually pouring into us when you see us as yourselves. And I think this right here is just like really will make that stronger. And I would like to connect with someone on the more social side of things to help me to explore that further. Okay. All right. At the end, this is in the north. This person here is African American. And at the end of the day, this is what it's about here. So where it's true that many African Americans and people of African descent don't know about, certain African cultures and histories and things like that, we also need you to help us to know it. Okay. And we also would like for you to understand the history of Ghanaians when they traveled to America for the first time. Okay. And how you've come to be African Americans today in the US. Okay. So then I'll also share with you the African American perspectives. So this was my started out as a dissertation project that has continued. So this is, I wanted to understand, I knew that there was some type of ethnic identity and family identity that was taking place. And I wanted to understand the processes of that. Meant to take this slide out, but basically the theory says that our identity is shaped by socialization, family, school, society, media, all these things go into shaping who we are. And so we got our personal exploration, what we understand, the narratives, the rituals, and our commitment is referring to those behaviors that we try out, try this, try that, and then what makes us us. If you don't mind, I will skip past this part, but I'm happy to talk about it more should you be interested in the social aspect of it. So I did use, this is a qualitative study, grounded theory. And we started with 7. We now have about 20 or 30 folks that we've interviewed. And just to, so this is the time of experiences of taking the test, getting the results from a commercial company, contacting the relative, and then continue to use genetic genealogy. I just want to share a few quotes with you so you can get, the perspective. I will let you know if these quotes are representative of the group or just a unique one. There were 3 things I found that were key: the sense of missing ancestral history, this involving African ethnic identity, and then recognizing relatedness or that family aspect of it. So I want to look here to the ethnic identity due to learning the ethnicity of the relative that they found from Africa. And it involved basically looking at the test results. And then at that point, so oftentimes when African Americans come into this, they're already looking at general African history. But then once they find a relative, then it sort of targets where they're searching. Okay. So this is Willie. He says, he took selfless time out of his schedule to just educate me on the Mandinka culture and his mother and his father. So again, he took the test. This is not a test that says you're Mandinka, right? This is a test to say this is a relative. You have some generations that's kind of close during a period of transatlantic slave trade period. So he connected with them, talked about their cultural history as well as their family history. Here, I still consider myself African American, but also keep in mind I'm Asante. The idea, knowing that my ancestors are from the Akan group, that brings me great pride, and both groups are represented. It's a little intensified. So whereas we use the phrase African American, now as you're finding relatives, the African takes on a new meaning when we're saying African American. It's actually representing certain groups and families. And then with recognizing the kinship, Nichelle says, this is generally a sort of a universal experience here. Most people end up going back to the phrase African American because you're finding relatives from various places across the continent. Here, I want to see my family because they are my cousins, and they're not just genetic cousins, they're my cousins. This one is a general. Again, once you find out that you are related, like the first cousin, second removed or whatever, tends to not be relevant. It's just they're my cousin. Then the first step is knowing that you're related, but the second step is developing those common interests. So it's not, just some artifact or something like that. They're actually trying to know each other as people, as what it could be. I'm not saying it's necessarily immediately family, but they make it what they want for themselves. Oops, sorry. Psychological impact as we wrap up. The well-being, acceptance, and belonging and pride was to be expected, but it was also changes in perceptions, phenotypical self-love, and this evolving generational orientation, meaning that before, I mentioned before that that the end of the slave period of trafficking was about 4 generations. So that makes it really challenging to know who your relatives are before then, because the people were listed as property, as numbers and things like that. So because of finding relatives, that generational orientation is changing because now we know more generations. And these are the last 2. This is a very dark-skinned man, African-American man, who, when he was growing up, he hated looking at himself in the mirror because people picked on him a lot, right? And so he he avoided looking himself in the mirror. And now, after finding relatives, after learning more about African history and things like that, he says, it's enjoyable to see myself. There's no pain when I look at myself in the mirror now. There's a degree of pleasure sometimes. Okay, that's actually your Senegalese side that you're looking at right now. Your nose look like just in something positive, not negative anymore. So that's Marcus coming to love himself because of his new family members he found. And then the other, to be able to scientifically state that I have genetic ties back to these particular regions, it's really influenced how I see myself and knowing that I'm not just defined by the experience of slavery. Yeah, you might be surprised at how much resistance people of African descent will face when we are saying that we come from more than, the slave ships and that we have family members. I mean, there will be people who in your face, you can claim that because we have separated you, and I don't know what it is, but, so this is revolutionary that we're doing. Okay. So in wrapping up, this has this deep-seated psychological impact on people. So yes, we are dealing with genetics. I know Professor Mills, you can't interview bacteria and things like that, but we're looking at genetics and we're making life changes here, and as you brought up, they're not all necessarily good. We have to, consider the ethical issues, but there is a lot that's happening here. So we are working on this website. It's under development right now. So you see a placeholder if you go to it, but this is the website. Many thanks to the University of Illinois, University of Cape Coast, and some other, folks that's on the team, definitely community members who started with me from the beginning in Paga. All right, thank you. Check it out. Thanks.
Okay, so I think you can have a seat, then you take some water, and then the discussion will start. And when there is the need for you to respond. But so colleagues, I can see you're enjoying the presentation. And as some of you were thinking how beneficial it's such a city. Yeah. If you've been to US and then you've interacted with the blacks, you see how important this is. But when it comes to the well-being of human beings, the most important aspect of it is the mind. And when it comes to the mind, but our mind is the contact of the spirit. So that is your conscience, the contact of the spirit. No. So I always tell people that you start getting sick from the mind. So your mind will sick. So that is the spirit. So for you to be healthy, one important thing is acceptance. Acceptance. To know that you belong to a family, you belong to people who accept you as the way you are, and then you share certain things in common. Very, 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 very important. So you go to US and there are certain blacks who do not know where they come from. They don't even know any cousin beyond their mother. So you are fortunate. If something happens, you can talk to an uncle here. You can talk to a grandma here, you can talk. They don't have that. So I will have a visitor here he is a Jamaican from Caribbean. But he's done a test and he traces ancestry to Ashanti region. And he's told that he was born on Wednesday, so he's told that Wednesdays the ones are called Kua. Yeah. So he has some bundle of this written on it. Yeah. You see it and you think, this woman is it, but that is acceptance you're talking. You get it. So it's very, very important to them and even to us. Sometimes your mother takes you to your hometown and introduces you to your cousins. See the way you feel. Although you are with your siblings, but seeing that this is my cousin. See the way you feel, that is acceptance. Okay. So this study will do a lot of healing than even a drug, which we think is the best way of helping heal society. But this study will do a lot of healing for people getting accepted, knowing that they belong to this family and that. It's very, very important. So the study is very, very important. So we are now going to open the floor. So thank you very much, Professor, for this presentation and an eye-opening. So we are going to open the floor for discussion. So I want you to also discuss to bring out beyond this, what are other things we can do with the data generated. And I know some of you are to help and others. So can we what do you call it? Maybe do searching information that can help in our area of expertise, that is the health with Muslim families. So it's very, very important thing. And I'm happy a professor of anthropology is here. We are finding it difficult to follow, but I know he understands this better. So as the discussion that I'm talking about the head of the anthropology. So as the discussion starts, and there's a way to maybe put us in the right way, is here to guide us. So colleagues, the floor is now open with the opening statement. We can now start the discussion. Thank you very much. So if you have a point, you are invited to share. Thank you very much. You didn't clap for me. Okay, so yeah, Dr. Alma, you are not a professor.
I heard something about you, that's why I'm coming. That's why probably this is the question. I would like to know, trying to locate the family, would there be a break somewhere where it will be very difficult to reach the lower parts? At times, when you go to the hospital, they misplace or they change. Do you think that you belong to this family? So, father, mother, don't share anything with them. Maybe at the top of things level, that person becomes a very great person in the family. But in trying to locate, then that big person is at the middle. It doesn't belong to where you start. In that case, how are you going to
Okay. So she's dodging the question, dodging it. So we'll take more to respond. Bob is also here to help too. I've forgotten the name. Dr. Brimbon is also here, so Dr. Brimbon also help us some of the response. So any other question? Programmer's question. So some big man in the middle want to connect so that you can benefit from the riches. You cannot be giving somebody who gives to you. Okay. Yeah, Richard.
In the genomics, we also do the and then we try to connect and find the answers. Yes. So for those of us who are in the first time, what you do is now applicable in the and for us, it may not mean so much the bacteria to say that you are connected to this people that is connected to you, they came up with what they said. And for this is very important. He also made mention of timeline. So you will between different, you will also have analyzing data and you will have to use some pipeline. How to write your own things. So in anthropology or in genetic genealogy, they also have their own things that we also in pathology and other use. But it's not so far from what we have studied. But the relevant is the social importance that is great. Supposed to be in Ghana, the people in the diaspora. So mine is just a contribution and 5% to the.
Thank you very much. Any further discussion, or maybe you can attempt to answer Dr. Ahmed's question, and I'm sure. Okay.
So my question, I will emphasize on the superiority of your technology. These are the commercial one. And what about the challenges?
Okay. Yeah, please you can answer.
Okay, all right. So in terms of superior or not, I wouldn't necessarily say that. But I do think that we have a more conscious about the ethical implications than the commercial companies do. And how they are indicating the results is very misleading. And that's something that we want to try not to do. And in fact, we're taking a lot of time in putting in education information that our participants, will see. So in that sense, I think we're more careful about it. The Python is machine learning. As Professor Neil said, you can actually adapt from other, uses to what we're doing. So I don't think in that in of itself is better, but the way that we care for and the questions that we're asking is for us. As far as the challenges, the commercial companies have a bigger net. They have millions and millions of people. They know people know them and people will want to test. So the success of this is the larger the pool of people that we have. And because they have so many millions of people who tested already, any random person who tests with them, they're bound to find a relative. Whereas in my case, right now, that's not necessarily true, which is why we have to be really particular in who we test. And it's not like it's a random family. So I would say really is the number of people that we have on our database right now, which I'm asking what we can.
Thank you. Please, you can also answer the first question, if you still remember.
Yeah, for lots of parts too. Maybe this was that. So I know you asked about the break point, but I would say that parent and offspring 100% of the time will bond that person if we happen to accept that person. First cousin, second cousin, we're likely to find, that close family member. So we will be able to, if we happen to have that person in. At a hospital, I imagine there's a larger database where that would be a lot easier for them to do. When you get to the fourth cousin and fifth cousin level, there's a chance you could be biologically related to someone and not show up in that test, right? So there's nothing else to do for biology other than test more people to happen to farm that doesn't, there isn't a way to really pinpoint if they go, like where we should start. And Professor can,
Okay. If you want to ask something. So, mine, I think is going to be completely different. You highlighted the significance of these projects, the social significance of this project. And I'm wondering whether there are plans to take this from academia to the public realm. In other words, like going to ATM, going to Peace FM, or going to God FM to explain the significance so that the public will get Yeah.
Okay. So, yeah, this is very much a public facing. So, us who are academics, like what counter promotion or what stuff necessarily. But in my university, they're changing to value this public engagement part, and I just came along at the right time. So yeah, the public engagement part is a very big part. We get a lot of institutional support for this. So in Ghana, we do have plans to do a radio interview once we get those details. And what I'm hoping, and I haven't really had a conversation yet, is to tie it into the emancipation celebration and potentially have a symposium or something every year. In the US, though, it is very much more public engagement. So our state representative for our area, State Representative Cheryl Adams, have passed through one of our houses, House Resolution 453, which is giving 900 people in our state a free DNA test to participate in this very project. This is a pilot study. In 2 or 3 years' time, her goal is for every person of African descent that lives in the state of Illinois to be able to have a free test. So that's very much public. There's also an article that was published in the National Black Caucus in the US. So now there's a nationwide awareness where she will invite me to give a presentation to the National Black Caucus to be able to take it elsewhere. So we already have, like, other than the state of Illinois, we also have New York and California, who is very much aware of this, to bring it directly to the people. So this is one of those rare moments where there is an academic study, but it has direct policy implications and community participation.
Thank you. Yeah, I want to ask.
We have a small house. So public engagement over ATL, but they communicate that because of the kind of here, and then we can have. But going forward, we want to include communities from we are think where the program the last part. So all the areas to choose this, we really need to make it. That's why public engagement. But we're going to have a there for the community. So we've already got some people, we can contact some people, so we can talk together. So also be glad we can work.
Yeah, so that is one area where scientists not done very well is dissemination. That is after study, and so it's good that now we want to involve a community, because usually our research should impact the community. Otherwise, then it's just for academic exercise with no impact. So it's very important. And then in so doing, mostly we should be able to classify our targets. And then if we classify our targets, you're able to know the specific channel of communication that will suit them. So for instance, we usually talk of radio. Yeah, you can broadcast on radio and it goes nowhere. It is the same people who read the article now here on radio. You get, and one thing I've also realized is usually when you go down to the community and then you involve the opinion leaders in the community, you train them, pass the information to them, and then they rather the people get it better than you when you do it on radio or on broadcast. Yeah. So and that is one thing I've learned from community engagement. So it's very, very important that you are taking that step. And I'll say that is one thing that our school is also very keen on. So we are looking at getting what they call it, a department or a unit to specifically focus on community engagement. So that most of the research we do will go down to the community, because that's where the impacts should be felt. And we do the research and then we keep it. We can also say get the promotions and all that, then the hypes, and hence then you get the harvest. In most advanced countries, the research impact is very important, and then the impact is felt at the community level. And if I talk of the community, it doesn't mean a village. Community is the specific people, what they call it, the study to impact. So it could be this level. You get it. So if there's a group in this level you want to target, it's a community. So when you talk of the community, it's a target group. So that is what we are looking at. So which is thing is very important. And the school is ready to help partner, push, especially the community aspect, and also the research aspect. The school is willing to support. Dr. Gatti.
So, my second question, my last question, error. What's the salutation to minimize errors in these mathematics?
Okay. So errors can come in at multiple instances. One of it is that's the way of being right? And so the first thing is to select appropriate array. The array that we chose is the one that's used in the US and the NIH, all of us project, we're using the same array. So we begin there. The lab is at the University of Illinois. They provide these services. So we want to make sure that we have a very low genotyping error. So assuming that part is outside of my lab. So assuming all of that when it comes to me, we do the bioinformatics. So same quality control. First of all, we have to make sure that it's staged properly. And that's part of the reason why we test parent and offspring. So it's a better way of phasing the data than to use the complication, in my opinion. Because so much of our reference panels are being conflict the European population. And I don't really trust that it will look at properly. So we try as much as possible to do parent offspring to phase it. So once it's phased, then it's just a matter of seeing, what's similar and what's not. And that is more or less straightforward. In terms of the predictions, we do try to make sure, to communicate that it is a prediction. But that some of the predictions have a greater confidence in the other. And I can't rattle off the numbers right off, but in the first 1 or 2 generations, it's more or less like very little. I won't say 100% correct, obviously a scientist would really say that, but it's very, very low. Once we get to about 3 and 4 generations out, we could be off by 1 generation. And once we get 1, 2, 3, 4, like 5 and 6, and 7, we could be off by 2 or 3 generations. Meaning that if I estimated that the common recombinant ancestor is 6, it could be 7 or 8 or 5. The more that we test individuals in that family tree, that reduces the range of where it could possibly be. So in this way, we reduce the inserted pre-analysis. I hope that answers your question. I can do a follow up.
Yeah, I'll come to you.
So my question is more on education. I see you portraying this as steady, clarity, as preparation kind of process. But I have this concern of who's. So it looks like it won't look good or it will favor come back down here. It's something to be right. For the average family that they are linked to. Subsequently, they become left on their own. These people do more rejected. So as you are looking more into the social, I think I want to raise your awareness of our end goals, accepting these people. It's not always like they have to bring something for the answer. But I'm a witness to a few examples. You know, as well. When a super good big brought, when the money they brought gets spent, then they get left on their own, thereabouts. So thank you very much.
We take some remedial measures.
So
I just have that you know, there's a couple of things there. When I'm presenting, I thought to make it a point to express that the community that we started out with is low income. And that this is not particularly representative of all of them. So for instance, the family that you saw there in the family on school and things like that. And so the interaction with Rhoda and her cousins, Rhoda's in Canada right now, she has a PhD and stuff like that. It's different, you know, it is not about, you know, these financial things, it's, oh, let's go on a trip together, you know, things like that. But in all honesty, you know, many folks, we learn about each other through the media. African Americans learn about Africa through the media and vice versa, and it's not good. And so it's really difficult to like really sort of communicate that. Yeah, so most African Americans will not be able to travel here and things like that too, right? And so we are trying to look at video conference because most people do have a smartphone, and to be able to do sort of a live TV, live conferences when we do travel. So it's not, you know, it's not talking to incidents. So we can have more and more conversation about that. So I do think it's really important to factor those things in. Not every person, not like on both sides. Not every African person that we reach out to will want to be related to an African person, and vice versa. But there's lots of it to deal with. But if I may, I want to tell this story. So one of the people that I quoted, his mother. So the son is very much into genealogy, and he found like several Nigerian cousins. But before he did that, he was telling his mom, you know, like what he was doing. His mother said, and I'm sorry, his mother said, like, I don't want to be related to your Nigerian. And you know, the son was not the therapy thing because it's what he wanted to do. So fast forward, he found some relatives, and he invited the relatives to the cookout, which is a really big thing in African culture. And when he did go, his family members blocked the cousin with all these questions and, you know, just walking around and stuff like that. So it seems to me that when it's hypothetical and the narrative within, oh, you'll never be able to find your relative, is more or less like, you know, some very ignorant, you know, responses to it. But when it becomes a reality, it's very different. When it's a cousin that you can, flesh and blood, it becomes a different thing in people's minds. Either, and I'm still exploring this, but either what they really wanted and how to come forward, or their moms changed. But you bring up some really important points. I don't want to like cover over any negative, issues, because again, we're doing some poor work here, and we have to be careful about it. So I appreciate you bringing that up.
Thank you. Okay, so with regard to,
I think very much. I have two questions. First of all, I realized you were using SNPs for the genotype because, and I'm thinking that with SNP, then you can have up to a maximum of 4 alleles to reproduce. Then when we focus on the microphenotype and haplotypes, we have a large variety of alleles. And by so doing, the margin of error would be reduced. And also, the second question is in connection with transatlantic slave trade. But in the slave camps, we heard stories of, we have some information on the fact that there was inbreeding and there was some kind of relationship, slave masters and slaves. So I didn't see some aspects of admixture analysis to tell these people how much of them is European, how much of them. And then also in cluster analysis, because we don't have so much reference from data, we tend to focus on Nigeria, looking at some clustering in Nigeria to do some GWAT studies that are relevant in as much as we are closely related in terms of social and lifestyle. And I think genetically, there is so much disparity, and if you're comparing our genetic information to that of Nigeria, then we are exposing ourselves to so many errors. So that in terms of linking the people in America to where they are assumed to originate, we don't actually miss much.
Okay, thank you.
okay.
I'm gonna come up here.
Yeah, you can. Yeah.
I respectfully disagree, but I'm gonna explain why. You're thinking of it from a population genetic perspective, and I appreciate that because that's the that's your field. But this isn't population genetic, right? So the first question you mentioned was about the SNPs. You said 4. Am I correct?
Yes.
Ancestry does 7,000 SNPs across the genome. The array that we're using is 1.8 million SNPs, right? So, that's an order of magnitude different.
So the SNP as a look at, you can have a magnitude of 4.
Not in that case.
Not the whole genome.
No. What I'm saying is, even it's like 1,000 some odd SNP here, and then we know that there's spaces in between those, right? So we're looking at relatively speaking a large amount. So even in the 4 SNP that you're looking at, if you go from the beginning and base here, your symptom organs, I probably assure you, is very, very small. Not even one properly. We're looking on the order of 8, 18, 17, which is very low. So any population genetic study that you're looking at in foreign operation periods, we're not even on the same, we're not even talking about really the same thing, because it's just so much smaller. So we, at our level, we're more refined, and we have a better representation of the genome. If you're looking at that segment, 1,000 small SNPs compared to 4 SNPs is a big difference, right? So we're going to be able to pick up more differences. And as you know, a SNP is the product. We know that it's going to be a different. We're expecting it to be a difference in at least 1% of the population. That's why we use, right? So we're using 1,000 of them, so our error will be reduced. So it's not even in the same magnitude. Okay. We are moving away from the word admixture, and I'll let it wrap up. You can explain more. how much a person have that, of their genome is because they're related to Europeans that are relevant to my study. The purpose of my study is to identify and the social aspect of it. I can run RFPs and the other tools that's out there to give a percentage of the genome that's associated with this particular disease. But that's not the point, right? There's also issues when you're saying Ghanaian DNA, Nigerian DNA, there's this idea that there's a distinct difference. When, as you know, if you're looking at, like, let's say, a set of 100 SNPs, right? Most of those are going to be the same. Overwhelmingly, majority, you already know that 99.9% anyway. And we really focus on a tiny bit of difference between one and another group. But first of all, the groups are the same. It was because of racism that the idea that the artist came up in the first place. But even when we're looking at that very small amount, if you're looking at one SNP difference between, like, a SNP, an allele, just happen to be in one group of persons than another, if you respond farther enough, you'll see it actually clustered in family groups, because that's how these SNPs get transmitted, right? Wait long enough, it's going to be another population as well. That's how mutations work, right? Also, when you're looking at the difference, it is more a matter of frequency rather than individual. And yours, you're looking at the group. The group, 80% have this particular allele, or so on, right? And maybe 60% have this allele. Not that it's different. Not that you have different alleles, right? It's that how many in that group, in that population have it versus the other. The reason why I'm emphasizing what you already know is because in the US, you have to emphasize the fight against racism. Really on our behalf. Okay? So you are correct from a population standpoint. But what we're doing, I'm emphasizing again, it's not population genetics. But even if we were doing population genetics, the framing of it is very misguided in terms of, like, what admixture means and these differences in groups and stuff like that.
Thank you very much. So I think, yeah, so your question was good, and when a question is good, it gets the right attention. Yeah. So so you was coming from a population genetics point of view. So even in Nigeria, what would I say? The population genetics across Nigeria will not be the same. But when you talk of the genome is the same everywhere. So what brings about the difference is the mutation. And there are a lot of things that go into the mutation. The reason why maybe, which I agree with you to some extent, maybe the genome here will be quite different from Nigeria. Has to do a lot of things that contribute to this mutation that brings about the difference. So from Adam and Eve, the genome should have been the same. If there were no mutations, we would have the same genome. You get it? So I agree with you that, but what they are doing is quite different in a sense. You get it? It's quite different in a sense. But what I will also say is that from here, maybe Rachel and I've forgotten the name. I'm not good at names. Yeah, so LaKisha, LaKisha. So from here, I think some of these questions are coming up because they want to get more from this study than what is happening now. That is what I get from. So they can get more from this study by just the structure of the questionnaire. the DNA sample will be the same, but the questionnaire, the kind of information you pick from the people, and we'll introduce different aspects, which different groups with different interests can also look at. You get it? So that is, I think that is where the reason why some of these questions are coming up. But as you said, your study is well structured, and then you know your target. But they are also, I could even see Rachel may be interested in even maybe other health aspects. So, for instance, if it comes to your study, one interesting thing that has to do with your studies, even when it comes to transplanting in an organ, you get that is in medical field. If it comes to what they call it, what is the common transplant? Because when it comes to marrow, bone marrow transplant and all that. So this, I remember about 15 years or so ago, there was a Black American gentleman who had leukemia, and then he was looking for. But I would say that is when this genetics actually started getting attention in Ghana. He was called something white. He was living in UK, and then he developed leukemia, and he wanted a transplant. He never had a match. He didn't get any match in UK, but he got a match in Ghana. I don't know, they did a transplant, all right, but he died. But at least he found a match in Ghana. You get that. So I still remember. And it was through this OD. You get that. So it's very important. So it depends on the questionnaire, how we structure the questionnaire. You can look at a lot of aspects apart from what we are looking at. So maybe as a Humphrey may be interested in population genetics as it relates to diseases, and then how now there's something we call targeted therapy and specific therapy and all that. And it has to do with genetics and all that. So this with the DNA material and then a proper structured questionnaire, a lot of information can be picked from the environment or from the people, and then a lot of analysis can be done alongside what you are doing. So I think that is where it's coming from. So thank you very much. I'm not sure there are further questions. Okay. So if there are no further questions, I don't know if I need to sum up. A lot of things have been said. I've been educated. I like attending such seminars. You will learn a lot, and you get a lot from it. And then the experts also tend to learn from even us sometimes. Based on some of the questions that come, if you go to the community, sometimes you go to the community with some information that is very good information, but interacting with them, sometimes you need to revise how you are going about the set. So it's always good to share. And so I will say thank you very much for coming. Let's keep discussing this. Rachel is here. And then David. Okay. Yeah, okay. So Prof. David is here. You from here you can still talk to them. And then for instance, I'm going to talk to them. And as I said, when it comes to the community, what they call it, engagement, it is something we are much interested in. And then I also want to pledge the support of the school. But we are ready to support you in any way to get to where you want to get to. So involve the school. Although I'm the team, I have the right to speak for all the team. So we are involved the school, and we will support you since you are working with biomedicals and this is under the school is ready to support you resources, human resource, material resource, and everything, because it's a good thing you are doing. So thank you very much, and thank you to everybody. I now hand over to the MC.
All right, so thanks everyone for joining this program. And I hope you all learned a lot. Some of us learned a lot. So grateful for your presence. We'll now take closing prayer and then we will spread. So let's pray. Almighty and everything, we thank you for how far you have brought us in this semester. We are most grateful that we are able to exhort whatever we intend to do. We are most grateful that we have broadened our scope and we have understood. And we have also understood the relevance of study in much advanced levels. We ask that whatever we have discussed here, you remind us to produce good fruit. This and many others should have fruit as our Lord. Amen.
Amen. Senior members, we want to take a picture, and it will be an evidence that something of this sort happened. So maybe the postgraduate students can join us. Well, let's all people, and we will take a picture. Thanks.
Yeah. Yeah. Where is? Okay. Zone, where? Oh, okay, so that one.